Alrighty, boys and girls, so now we are going to get started on Unit 5. So on Thursday of last week, you took your quiz on Unit 4. You got those back today. The grades were very good. I was very impressed with you guys. So now we're going to move on to Unit 5. In this unit, we are going to be talking about genetics and heredity. This is a really fun unit because it's all about why you are the way you are. So it's all about you, okay? So let's go ahead and get started. Now, if you've answered your warm-up, so the bell work for you guys, the warm-up for you guys this time was, in what organelle do we find the instructions for our traits? So why do we look the way we look? In what organelle do we find the instructions that make us look the way we do, that make us act the way we do? Okay? And that is our nucleus, right? Okay? And the second question was, what are those instructions called? And if you guys knew the first part, then you're guaranteed to know the second part, which was DNA. Good. All right? Next, let's move on. So, if you were in class on Friday when everybody else was having fun at Defy Gravity, then you know that we watched a video about the history and the structure and the function of DNA. It was a pretty long video, but it was very cool because it walked you through exactly how these scientists came upon the structure of DNA. And if you're thinking about the correct scientists, and you were thinking about Watson and Crick, they were indeed the scientists who discovered the structure of DNA, or who put the pieces together, because it was really a culmination of everybody's work. So we're going to really quick just go over that and start at the source for those of you guys who were not here. DNA is also known as deoxyribonucleic acid. Okay? And it is in the nucleus of every living cell. You guys already knew that. DNA was discovered in 1869, but the structure was kind of a mystery because they didn't really have the technology to figure it out, and no one knew really where to start. Okay, but they knew that it had to be something simple because DNA is passed from every generation to generation. So they knew it had to be something simple because otherwise it wouldn't be able to be passed from generation to generation. So in 1952, using her X-ray photography, Rosalind Franklin, who I absolutely adore and love, observed that DNA could be identified in the shape of a helix. Because what she did was she took X-rays or pictures using X-rays of DNA. So she extracted the DNA from cells, crystallized it so it was really hard, and then put it inside an X-ray tube and shot X-rays at it. And what that did was it kind of like made shadows on photography paper, and that's how she was able to figure out that the shape of DNA was in that of a double helix. Or a helix, sorry, not a double helix. But she couldn't really identify the shape of it until a year later when Francis Crick and James Watson used her images, which they got from Maurice Wilkins, to figure out that it was in the shape of a twisted ladder, okay? And that twisted ladder, if you imagine a ladder, okay, and then you twist it kind of like a staircase, that's what the shape of a double helix is. So DNA's structure, how we describe it, the shape of it, is a double helix. Okay, so if on a test I was to ask you what the shape of DNA was, its shape would be a double helix. All right, so quickly, let's go back over this. DNA is known as deoxyribonucleic acid. It's in the nucleus of every living cell. It was discovered in 1869. Rosalind Franklin was the first person to put a piece into this puzzle when she used X-ray photography to take pictures of DNA. Unfortunately, she could not identify the shape. And then a year later, in 1953, Francis Crick and James Watson used her images to describe the shape of DNA as a double helix. Okay? Moving on. So the structure of DNA is indeed a double helix. And like I said, you can imagine it like a twisted ladder or a staircase. So if you take a ladder or a staircase and then twist it, that's what a double helix looks like. Now DNA is made up of three things. Okay, it's made up of a sugar or deoxyribose. That's what the part of the name means. So that's the sugar part. A phosphate, and then a nitrogen base. Okay, and you've got 4 nitrogen bases. You've got thymine, adenine, guanine, and cytosine. And they all match up together, and we'll get into that in just a few minutes. But DNA is made up of nucleotides, and then these nucleotides are made up of these 3 things: the sugar, the phosphate, and the base. Okay, you guys need to know that. DNA is made up of nucleotides, and nucleotides are made up of sugar, phosphates, and bases. Now, if you look at a picture of DNA, and if you just lay it out flat, like a ladder would be laying out flat, if you look at the sides of the ladder, okay, the sugar and the phosphate make up the sides of the ladder. The rungs of the ladder, or the part that you step on, is the bases. And then those bases are joined together by hydrogen bonds. Okay? And if you guys think back to when we were talking about water is held together by hydrogen bonds. And remember I said that hydrogen bonds are really weak. That's what makes water easily to change state, to go from a solid to a liquid to a gas. It's because it's joined by hydrogen bonds, and hydrogen bonds are weak. So this is what makes DNA easy to unzip and then replicate and put into those cells so it can be passed from generation to generation to generation. It's because the middle of it is made up of these hydrogen bonds, and these hydrogen bonds are easy to break apart so it can quickly unzip itself and replicate and then be passed from generation to generation to generation. Okay? So very quickly, let's go over this again. So DNA structure is that of a double helix. DNA is made up of nucleotides. And then those nucleotides are made up of a sugar, a phosphate, and a base. Okay? The sugar and the phosphate are the sides of the ladder, or are the sides of the double helix. And then the rungs of the ladder, or the flat part of the double helix, are the nitrogen bases, and they're held together by hydrogen bonds. All right, moving on. Now, like I said before, the steps of the ladder are made up of the pairs of molecules called the nitrogen bases. And I said that there are 4 kinds of nitrogen bases, and you guys are going to need to know these. Okay? The first one is adenine, thymine, cytosine, and guanine. All right? Adenine only pairs with thymine. So A only pairs with T. That's it. Cytosine only pairs with guanine. That's it. C only pairs with G. You can't make it any way else. That's it. Okay? Now, in 1950, how they found this out, that A only pairs with T and C only pairs with G, was in 1950, Erwin Schargraf analyzed the base pair composition of DNA, and he found out that the percentage of adenine matched up with the percentage of thymine. The percentage of cytosine matched up with the percentage of guanine. And this told him that what you have of adenine, you have to have of thymine. What you have of cytosine, you have to have of guanine. There's no going back and forth. You have to have the same amount of adenine as you do thymine, the same amount of cytosine that you do guanine. This means that there is the exact same amount of adenine and thymine and the exact same amount of cytosine and guanine in each base pair. That's it. Nothing else. All right? Now, since we've done this and we talked about how they base pair with one another, what I want you to do is take your worksheet that I gave you. It's called We're All Peas and Carrots. Okay? And what I want you to do is I want you to match up those base pairs. So in front of you, you have boxes with A's and T's and G's and C's in them, and I want you to match those up with their base pair. You've got 2 of those to do. I want you to do those right now. All right, and then after that, what I want you to do is take your other worksheet Okay, it's called DNA, the double helix. And I want you to color code the bases, okay, and then match those up on the rungs of your DNA double helix, okay? So if it says you have a T and you need to color it red, okay, then you need to have a T, then you need to color that T red and then match it up with your A that's, I don't know, yellow, okay? You need to match it up with that one. All right, so that's what I want you to do next, those two worksheets, okay? And you need to cut those out and glue those into your notebook so that you'll have practice from doing this. And then after that, you can come to me and you can get your homework, okay? Which is doing the exact same thing, you're just getting more practice with it. Alrighty? So this was all about the structure and the function of DNA. We did a little bit about history. That wasn't a whole lot. The main thing was the structure and the function. So the structure is the double helix, and the structure really accounts for the function. Okay? The structure is it's easy to unzip. It's easy to pass from generation to generation, and that's its function. Its function is to pass those traits, to pass those genes on from generation to generation. So the structure really dictates the function of DNA. All right? That's what we talked about today. Get started on those two things. When you're done with that, you can come up to me and get your homework, and you can get started on that. Alrighty. I will talk to you guys later.